good afternoon, good evening, everybody. Welcome to the ReSViNET webinar. It's good to see you all. My name is Bont, and I'm a pediatric infectious disease specialist and the founding chairman of the ReSViNET Foundation. And on behalf of the foundation, I welcome you to our webinar today. After a few introducing slides, we will immediately start with the webinar of today. Our vision is to connect global expertise and leadership in order to decrease the global burden of RSV infection. And our mission is to support performance of high quality research, to improve knowledge of RSV epidemiology, which is what we're here for today, and to develop safe and effective therapeutic and preventive interventions. And I think we're all very excited of all the developments in the field at the moment. And our goals are to advocate and create awareness for RSV, to act as a focal point for effective partnership with stakeholders, and to combine knowledge and capacity required to enhance development of novel RSV therapeutics. So why do we do RSV research and education? There are 33 million patients, 3 million hospitalizations, and at least 120,000 deaths, most of them occurring in the first months of life. For our educational mission, we have developed a webinar series to promote knowledge sharing of the latest developments on publications, on groundbreaking studies, and to provide education to professionals, young professionals in the field. We organize our webinar series every 3 months, and we look back on 13 webinars so far. The average number of participants is about 120, with good ratings, very diverse global representation, and good, well-evaluated interaction. Oh, sorry. So every webinar is followed by a specific webinar presented by Dr. De Frankrijker, who is translating the webinar for professionals for the general public. And also the webinar of today, RSV Travels by Airplane, will be broadcasted January 26th. Usually we do a shorter time interval, but for the holiday season, practical reasons, there's a little bit longer time interval. So before we start with the webinar, I want you to be aware that we're very proud to have our next webinar in Mumbai, India, because our RSV Vaccines for the World is really meant very literally. We are there to get the vaccines everywhere. And Mumbai, we have a fantastic venue, and we encountered a lot of welcoming people in India. So please feel free to join our conference in 3 months. So the conference has 8 sessions on the global impact of RSV, patient involvement and ethics, clinical interventions and their global use, implementation of RSV interventions, clinical and secondary effects of RSV infection and prevention, vaccine immunology, low and middle income country health, economics and policy, and finally the global health challenges. We have master classes and young researchers tournament, really to show that we want to prepare the next generation of RSV researchers. There is a abstract submission deadline, December 1st for poster presentation, and for companies for oral and poster presentations, we have a late breaking deadline of January 15th. And again, this is what I already told you. There is a website that you should feel free to get further information. So without further delay, the coming 20-25 minutes, Professor Philippe Lemey from the Laboratory of Clinical and Epidemiological Virology at the Riga Institute in Leuven will explain to us about novel insights of RSV molecular epidemiology and how that led to this very tentative title that RSV travels by airplane. Please use the Q&A button on your screen to ask questions during the meeting so that I can ask these questions following the meeting to Philippe. We really enjoy interaction with you, so please use that button as much as you can. Philippe, without further delay, the floor is yours.
Thank you, Louis. Let me see if I can share my slides. I think you should be seeing my screen.
Perfect, just perfect.
Great, thank you. Yeah, thank you for the kind introduction, the nice sketching the context of this work, and also for the opportunity to contribute to these webinar series. It's a real pleasure. So today I'm going to talk about a study that has been in review with Nature Communication, and I believe we're waiting for the final formal acceptance of the manuscript, and we hope that it will soon be available to the public. It's a study that was led by Anne Fleur Langendijk, who was recently awarded her PhD degree in Louis' team at the University of Utrecht. And the study involves all the partners of the INFORM RSV study, and they're listed here in small font with affiliations just to be able to fit everyone. Why am I presenting this work today? Well, a postdoc from my team, Bram Vanken, and I got involved some time ago to contribute essentially to the analysis methodology in the study. So we don't have an extensive expertise in RSV, to be honest, but my team at KU Leuven works on the molecular epidemiology and of different viruses, and also on some of the methodology to study their evolution and epidemiology. And it's this methodology is centered on the concept of phylogenetics and all the statistical modeling and inference this involves. With that work, we basically complement a large body of virology research that is done at the Reg Institute, which is an interdisciplinary biomedical research institute here in Leuven. Okay, so molecular epidemiology in broad terms, or genomic epidemiology, as we can call it nowadays, has the objective to understand how pathogens, and in our case viruses, transmit in the human population. And there are a number of different questions that one can ask about viral transmission. For example, how many introductions is a population or has a population been experiencing? From where are they being introduced? Is it from a single location, or does it come from multiple locations? Do these different lineages then show perhaps different transmission dynamics? And then for seasonal viruses, which is what we're interested in this webinar, we could ask ourselves whether this is really a repeatable pattern, as we perhaps could expect for sourcing dynamics as we see, for example, for influenza. Now, all of these questions can be addressed using genomic data by applying phylogenetic and phylodynamic approaches. And as we have witnessed during the COVID pandemic, this type of work can be important to provide a kind of a continuing understanding of virus evolution and the way it's spreading. And I think for RSV, this is only going to get more important with the application of immunization programs and potentially the spread of escape mutations that this may bring about. Now, the rapid evolutionary rate of viruses, and RNA viruses in particular, is really key to providing the information that we need to study their evolution and epidemiology. So RNA viruses generally evolve in the range around 10 to the minus 3 substitutions per site per year. And we can see that RSV genomes evolve perhaps a bit faster than your typical arbovirus, but at about the same rate as coronaviruses, but maybe lower than Ebola virus and influenza here. However, in terms of the time it takes to accumulate substitutions, and this is essentially what is shown here on the right, RSV is lagging not only Ebola, but also these coronaviruses. And the reason is because coronaviruses have longer genomes, and this provides, of course, more opportunity for substitutions to accumulate per unit of time. So this is also why working on complete genomes is really crucial. Now, if we would focus only on the attachment G gene, which has been done so often before in the field, we see, of course, a faster rate for that gene, but due to its limited size, it takes about 4 times longer to see substitutions accumulating. So complete genomes provide much more resolution to study molecular epidemiology. Now, there has been a rich development of statistical tools to extract epidemiological and evolutionary information from viral sequences. And in our study, we made extensively use of a Bayesian statistical framework called BEAST to perform these so-called phylogenetic analysis. So BEAST is centered on a phylogeny, but also includes other models, such as a molecular clock model to estimate these trees in time units, coalescent models to estimate how viral population sizes change through time, and then also these what we call diffusion models that allow us to do phylogeographic reconstructions, and I'll come back to those specifically later in this talk. Now, molecular epidemiology has been widely adopted in RSV, including sometimes also applications of this BEAST framework, but mostly at the local level. And as an example here, I'm using a study led by Charles Aguti and James Noak in Kenya, where it was shown, for example, that most of the circulating variants are detected only in a single season. And there are several new variants being introduced each epidemic season. Now, unfortunately, we lack good insights what really happens at the global level. So unlike for influenza, for example, which I already mentioned follows or is expected to follow, for example, this source-sink dynamic, and for which we have fairly good estimates of the degree of persistence versus new introductions seeding seasonal epidemics for the different subtypes. Now, this is where the Inform RSV study comes in, and this is really why it's so important, because it offers for the first time a systematic global sampling to start to look into this. And Louis could, of course, summarize this much better than I do, but Inform is a prospective global clinical study that includes 19 hospitals in 17 countries, where the sampling started from 2017, and it targets RSV positive children less than 5 years old, with a target of about 50 to 100 samples per site that are then sequenced using high throughput sequencing techniques, and for which consensus genomes are arrived. And below you see here a graphical summary of this sampling, extraction, amplification, and sequencing protocol that is being managed by Robert Jan Lebbing. So the primary aim of the study is to investigate the molecular heterogeneity of RSV isolates in this global population, and to really provide a repository of complete genome sequences for the community. And here's a graphical summary of that genomic data that has been generated in this case for samples prior to the COVID-19 pandemic. And this is what we focused on in our study. So this data was then further complemented with available RSV genome data from GenBank, which resulted in data sets of over 1,200 genomes for both RSV-A and RSV-B. So what we see here are the time-measured trees, so trees scaled really on a calendar time scale for those two subtypes, with tips colored according to the continent of sampling. So we had two main objectives in studying this data. The first one, to characterize the selection pressure across the genome and test whether we see turnover of the virus population and whether this is impacted by selection, and thus whether this deviates from sort of the neutral expectations of evolution. And the second goal was to explore predictors of global circulation and learn about what is really driving RSV transmission dynamics. Now, the first goal is somewhat the low-hanging fruit, because such aspects have been studied before for RSV. But here we had the opportunity to look into this with comparable and reasonably large-scale data sets for both RSV-A and B. So as expected, using codon substitution methods to measure what we call site-specific non-synonymous over synonymous substitution rates, we identify positive selection predominantly in sites in the attachment G protein, and in similar numbers or similar proportions in both RSV-A and B. We also find an occasional positively selected site in the F protein, and then a few in the polymerase, again with good consistency across the two subtypes in terms of numbers. Regarding that population turnover, we essentially measure this through three tree-shaped statistics. And I'm not going to go into this, but we essentially compare these statistics for the reconstructions from our genomic data to the ones that we can get from simulations under neutrality. And this is essentially what is shown on this graph for two different statistics. The big blob here is the neutral expectation for both RSV-A and B genealogies. And we see that what we get from our actual data deviates significantly from that neutral expectation. So we can conclude that both RSV-A and B are shaped by non-neutral population turnover. Now, concerning the second goal, which is new to the field of RSV molecular epidemiology, I would like to briefly or conceptually describe the approach that we took to confront this. So for this, we need to go back to our BEAST framework for phylogenetic analysis, and specifically to these discrete diffusion models that we can employ as part of the BEAST probabilistic model to perform phylogeographic analysis. So these models simply describe a process of transitioning between discrete locations across the phylogeny. And this is parameterized in terms of pairwise rates of transition. So basically the frequency by which viruses jump from one location to another. Now, what we can do is make those rates a simple log linear function of a number of potential predictors. And such predictors could simply be geographic distances, as illustrated on the left here, or it could represent demographic or mobility information. And as part of this model, we estimate the effect size of the predictors, these betas here. So these are standard contributions or effect sizes in GLM models. And we also include these deltas here, which are basically Boolean indicators and allow us to estimate the inclusion probability of predictors. So this allows us to really identify which of the whole set of predictors that we can propose explain the pattern of transitioning across the phylogeny. Now, if we apply this approach to our global RSV A and B data, either with countries or with continents as locations, continents on top here, countries at the bottom, with a number of basic predictors based on demography, population, so population sizes here at the origin and destination, geographic distance, this difference in latitude to account for seasonality, and air travel. We see that air travel essentially emerges as the strongest and the most consistently supported predictor in the model. So this is the case for both RSV A and B, and also if you apply the same model to both data sets jointly. And we see the support, whether we include in the sample sizes in green here or not, which is always a confounder for these type of analysis. So our sensitivity analysis indicates that the support for air travel is kind of robust to that. Now, interestingly, if we apply the same, if we apply a time inhomogeneous model, so allowing for different effect sizes, different contributions for the early years of sampling versus a period that largely includes the informed sampling, we see that the strong signal basically derives from the recent period of sampling that contains most genomes, but it's essentially also the period that is covered by the informed sampling. So it essentially demonstrates that we need substantial systematic sampling to get meaningful signal from the data. And that is really what inform is offering us. Now, what does this insight have to offer? Well, first of all, it might not be such a surprising finding, as air travel has been shown to be instrumental for influenza global circulation, for example. And also for COVID, this has obviously been important. But the fact that we find strong signal for this predictor implies that transmission through air travel is a relatively frequent event. And this is also in line with the degree of phylogenetic mixing that we find for genomes by country. And this is what is shown here using a measure of entropy, which is high for most countries. So suggesting that there is a fairly dynamic pattern of spread every seasonal period. So this knowledge can be useful when modeling the dynamics of spread, including that of potential escape mutations. And the ability to incorporate such mobility information as we do in this approach will also be important for future reconstructions of the spread of RSV. Now, what are the future directions for this type of work? Well, we have identified mobility as a driver of global RSV spread, but it would now be very useful to be able to also determine the actual patterns or the actual dynamics of spread. For example, do we expect source sink dynamics as was identified for flu, or do we expect to see a more generic sort of metapopulation dynamic model? In addition, I think it would be very insightful to also quantify the degree of persistence versus new introductions in seeding seasonal epidemics. And with genomic sampling of after the COVID control period, we would also be able to evaluate how this pattern has been affected by the control period, the control measures, and how this has affected the global RSV diversity. And finally, perhaps we can further improve on air travel as a predictor using model-based predictions that incorporate travel aspects, but also other types of epidemiological information. And some ongoing work with collaborators at INSERM in Paris seems to suggest that this is in fact the case for flu at least. So maybe we can also extend this type of work to RSV. Now, there's a lot of people that I would like to thank for this work and their involvement in the study. Of course, the study participants, their families, and the investigators, the research staff from Inform RSV, Outsmart RSV, and the South African pilot study laboratories who contributed to the data analysis. ReSViNET, that Louis nicely introduced, his whole team at Utrecht, all the local investigators who have made this work possible, and then of course the sponsors. So this work was supported by AstraZeneca and Sanofi, and Inform RSV provided logistical support through Julius Clinical, a founding partner of ReSViNET. And then also my sort of long-standing collaborators in molecular epidemiology. And with that, I would be happy to take questions. Thank you.
Thank you so much, Philippe. You always make it seem so easy to analyze and understand these molecular virology data. While people may have questions for you, I will start. So this transmission by air traffic, does that mean that RSV basically comes in the first cases and the local epidemic starts in highly populated airport-rich regions? Is that what you would expect? And that the direction of local spread is from airports to more rural places? Is that something you would expect based on this data?
Yeah, good question. So indeed, the frequency of spread will be determined by sort of the passenger flow between airports. So indeed, if the virus is being introduced somewhere, you would think that is indeed in an airport serving a larger city, and then it will diffuse to sort of more rural populations. That's kind of the expectations, and many models that try to look at spatial spread, like the GLEAM model by our partners from INSERM, use this type of kind of hierarchical mobility data. So you have sort of the global air travel, but then at a more local level, you have sort of more commuting type of data that kind of drives the spread.
Thank you. Thank you. And another question. So it appears you said, well, I hope there are some questions from the audience, people with a more molecular background, perhaps, epi background, perhaps. So you told me it is not really a surprise what we found, because we have also seen the same transmission pattern for other viruses, in particular mentioned influenza. Do you think that this is true for all these and COVID you mentioned, obviously? Do you think this is the case for all the respiratory viruses like human metapneumovirus, parainfluenza virus, adenovirus? Is this just like the way it is spreading? All these viruses have seasonality, or is there any reason to think that there might be viruses that have a different transmission pattern, in which maybe air traffic is not so important?
Yeah, I do think it's a fairly general process for most respiratory pathogens. I mean, viruses move through us, right? And we travel through airplanes. We also do not travel in sort of a distance-dependent manner across the globe. We jump on airplanes and are able to travel large distances. So this is what what our human viruses or human pathogens use as well to move across the globe. So I do think it's a fairly general pattern. But then again, it would be nice to stumble on some that actually do not seem to follow this pattern.
Yeah, you would have to confirm it for every new virus probably, but not every virus is so frequent. These are very, very resource-intensive studies. So I just thought maybe we can learn something general. So apparently, maybe this is a general phenomenon. So David Spiro from NIH mentions that RSV is often thought of as a virus that is transmitted by children, but this suggests that there is a large role for adults in RSV spread worldwide, so long distance spread, if you will. And he suggests, very speculative, but thank you for doing that, but maybe adult vaccination may be a way to decrease global spread. Any thoughts from you?
Yeah, that's a very good point, and it's actually an aspect that could make it slightly deviate from sort of a predominant air travel based dispersal pattern, because we know that it's, you know, the largest proportion of air travelers are adults, really. You know, there's very little infants, or at least the proportion of infant traveling over air travel is quite restricted. So this does beg the question, you know, indeed, is it primarily adults that are able to transmit the virus? Perhaps even, you know, adults that are asymptomatically infected, right? We know that this occurs. They're less likely, I think, to transmit the virus, asymptomatic infected people. But if they're quite frequent, then this could still be a mechanism by which the virus is spreading. So I think it's a very good point and something that deserves further attention, really.
Yeah. James Noakes from Kenya, I think both of us know him. For obvious reasons, he asks us whether there are plans to include locations from tropical countries in the INFORM study, which is indeed an ongoing study, and we intend for many more years to continue this study. And I will take that question, James. We find it very interesting to do that, and discussions are ongoing. I know that it is the ambition of the sponsor to make a global impact with infant immunization, but for many reasons this is still under consideration. But thank you for making that, for speaking up for the Global South. So this is really well appreciated. Are there any more questions from the audience? Oh, there are more questions in the Q&A. Thank you for pointing that out to me. I had missed that, sorry. So Kalina Zlateva, she thanks you for your presentation and wonders whether you did an analysis revealing significant differences between the transmission dynamics of RSV A and B. So you mentioned similarities, but did you find any differences in the dynamics, Philippe?
Yeah, good question. Hi, Kalina. It's good to see you out there. I've worked with Kalina, it's almost 20 years ago during her PhD here at the Rega. And I think it was the first time I was introduced to RSV. And we actually did some of the first sort of positively selected sites in the G protein at that time. In terms of sort of the drivers of spread, they were very similar. They were both predicted by air travel, so there weren't any big differences there. In terms of positively selected sites, those were similar in proportions across the three genes that I pointed out, but they were not necessarily in the same position. So There's different sites that are being targeted in these different subtypes. Also in sort of population turnover, RSVB is a bit stronger in turning over than RSVA. So they were both significantly deviating from neutrality, but it seemed that RSVB was doing this a bit faster than RSVA. Whether that is really sort of a general truth for these subtypes, I think remains to be shown. I think we need sort of a more extended sampling to see whether that will hold.
Thank you. Yes, we have thousands of more patients and samples to analyze, so the confirmation may come. So then Annabelle Rector, you know her very well. She is. I'm very glad, Annabelle, that our network is expanding and that people from other fields take an interest in RSV molecular epidemiology. So thank you for being here and making this important question whether the restriction to include only patients under 5 may have introduced a bias in the data set, Philippe. Your own colleague is now scrutinizing our work, so that's good.
Yeah, that's a very good point. Thank you, Annabelle. To be honest, I'm not sure how to answer that. Do we think that there are other variants circulating of RSV in other age groups that we would miss if we sample children less than 5 years? I wouldn't expect so, but then again, I don't have the data to really demonstrate this.
So the only thing I can mention is that, I have the same question, but that if you look at local epidemiology, there is some evidence, unconfirmed, but that the adult epidemic starts a little bit later than the pediatric epidemic. So there is some evidence that actually the first cases, the first wave of RSV start in children, and then 2 weeks later is the adults. So maybe there is indeed some differences in spread, but not so much in genetic composition of the virus, obviously. Yes. So then we have a question of Rahul Sharma. And there is a question that I only can see part of the question, so please add if you will, which is, do you think there is a preference for RSV spread based on genetic diversity? And is there any variant that you think is important here? That can be used as a genetic marker for a hotspot. Philippe.
Yeah, it's a challenging question. Haven't really thought about whether differences in genetic diversity could play a role. We applied the model to the entire population. I mean, if there would be variants that would be more transmissible, then those could be important in sort of the population turnover that we kind of measure from the genealogies. And those might then be more important, that those might also be important in sort of the global dispersal through air travel. But I haven't really seen like strong, let's say, directional selection where a particular phenotype really is beneficial and then basically takes over. Especially, for example, if you look at the trees and then the RSVA trees, there's quite a number of co-circulating lineages. So I don't expect that a particular SNP gene or a specific variant has been tremendously beneficial for transmission. But again, I think this is also something that requires further attention.
Thank you. So I think if there are no further questions, I would like to thank you, Philippe, for a great talk. I see others confirming that. I also want to thank all our collaborators, or a few of them that I can see here in the audience, in particular, Dee Wilkins. Glad to see you. And I hope to see you all soon. Again, we're going to translate this webinar for the general public. Our webinars are going to be available to others through our website, so you can actually review it or advise others to review it. And again, Philippe, see you soon. Everybody stay safe and hope to see you in Mumbai. Bye-bye.
Thank you. Bye-bye.